Hello everyone, I'm Azadeh. I'm a principal data scientist at Genentech, and today I will be presenting a work in collaboration between multiple teams of data scientists, clinical operations, and evidence generation. My goal today is to introduce our work, which is a path towards an automated and inclusive clinical research. In this talk, I will briefly introduce clinical trial matching and then share some very high-level information about our data and modeling challenges and our NLP model. Let's get started. We are all aware that medicine today is imprecise. Millions of people are taking medications that unfortunately will not help them. Top 10 highest grossing drugs in the United States only help 1 in 4 of the people who take them. There are also drugs that are harmful to certain ethnic groups because of the bias towards white Western participants in classical clinical trials. If we want to make clinical trials smarter, enable precision medicine and value based care, and to be able to deliver individual patient level medicine, we need to consider genomics, lifestyle, preferences, and health history. This means that for quality healthcare, we have to be able to process so many different sources of information where majority of data is unstructured and difficult to analyze and growing rapidly. According to research nowadays, doubling time of medical knowledge is only around 73 days compared to 50 years in 1950. This means to enable smarter clinical trials, we have to process and analyze more than 400,000 study protocols in clinicaltrials.gov or search for relevant contents from more than 35 million citations in PubMed, or process information of more than 1.2 billion unique non-identified patients in IQVIA or any other sources of claims data. Knowing that we have more than 4.5 million medical and healthcare concepts in UMLS, Unified Medical Language System. Humans cannot scale well when dealing with this large size of data. Hence, it's necessary to use some automated approaches. Natural language processing, or NLP, can help with this automation. NLP describes the interaction between human language and computers. It's a technology that many people use daily and has been around for many years, but it's often taken for granted. We have seen examples of NLP in action when using spelling and grammar check functionalities, or when we are using digital voice assistants, or simply searching Google or clinicaltrial.gov for a specific medical related concepts or study information. Clinical operation team members in their daily work might be using search functionalities of clinicaltrial.gov quite often. However, still they need to review hundreds of study protocols and consequently some relevant publications to utilize some insights from previous studies in their work. Where NLP outperform humans is in the amount of language and data that is able to process. Therefore, its potential uses go beyond the examples we see here and make it possible to use on tasks which can take humans months or years to accomplish. In the next set of slides, I will be focusing on introducing one of these tasks, which is called clinical trial patient matching. Clinical trials with an annual market of over $46 billion play an important role in drug development, but they often suffer from expensive and insufficient patient recruitment. Many trials struggle to enroll the required number of patients. Around 50% of trials are delayed due to patient recruitment issues, and around 25% of cancer trials fail to enroll sufficient patients. The recruitment cost is also high and estimated around $6,000 to $7,500 per patient, while there is no guarantee that a patient stays with a trial through the whole procedure. Considerably more concerning, the participating patients are not necessarily a good representation of the real world patient population. For example, 13.4% of the US population is Black, while only 5% of trial participants are Black. This is even worse for Latinx population, where 18% of the US population is Latinx and only 1% of trial participants belong to this ethnic group. As of today, for site selection, clinical operation teams work in collaboration with data owners to locate high priority sites that can enable faster enrollment given a clinical trial study protocol. This procedure is manual and rule-based with a focus on centralized site location, where data owners convert the natural language of inclusion and exclusion criteria from clinical trial study protocols to some query language to be able to identify providers of potential eligible patients. And this procedure can take at least 2 to 4 weeks per study in the best case scenario, and it can vary drastically depending on the scarcity of disease. Now, given all the stages of a clinical trial, if we can automate a portion of this procedure with the focus on clinical trial matching using AI NLP functionalities, we believe we would be able to inform study design and empower outreach programs. In other words, we can help with two stages of protocol design and site selection. So now let me explain in more details what is this clinical trial matching and why we think it can empower outreach programs. There are two common ways that the literature refers to patient trial matching. In the first definition, we know the patient and have access to their EHRs, and we are looking to find one or more clinical trials that this patient is potentially eligible to participate at. To our knowledge, there are more research focused on this problem. In the second definition, the one that we are targeting in our work, we have a clinical trial study protocol and we are looking to find eligible patients. However, the challenge in that is in all the data sets out there, patients are anonymized, meaning that in this definition, we don't have access to patient information anymore. Hence, the commonly known approach is using medical claims data and structured EHR data that is available in claims to find anonymized potential patients. And then, given that claims data have providers' information, we can simply identify providers of potential patients. And this information can be used by our clinical operation team to empower targeted outreach campaigns and to be able to educate providers of ongoing and upcoming clinical trials. The current data-driven methodologies, which mostly focus on centralized site location, are generally manual and rule-based. In these procedures, a clinical operation team member we'll be taking a designed or in progress clinical trial study protocol to data owners. And then these data owners start a time consuming and resource intensive procedure to convert each of the inclusion and exclusion criteria to some query language. And with this approach, they can identify anonymized potentially eligible patients and consequently their providers. Unfortunately, these methods are quite inflexible due to inadequate rule coverage, especially when it comes to rare diseases. So we just clarified what is the clinical trial matching we are referring to, and let me just briefly walk you through the procedure that we are working to automate using AI NLP. In claims data, we have some of the patient's demographics such as age and gender. We also have a list of procedures, diagnoses, and prescriptions from different claims records for that specific set of patients. And we can use those information against inclusion and exclusion criteria of a clinical trial to identify if a patient is eligible or not. And this way, we can compile the list of anonymized patients and then use claims data to identify their most relevant providers. This procedure can be automated using natural language processing models, and with an automated system, we can empower outreach programs as described earlier. We also believe with an automated system, we can inform clinical trial study design and evaluate the impact of changing inclusion and exclusion criteria on potential patient population, and consequently enable faster design and site selection cycles. We also believe this automated system can be of huge benefit if a clinical operation team is also interested to check viability of decentralized site selection. So concretely, the NLP engine will be looking at the natural language of clinical trial exclusion and inclusion criteria and also structured EHRs that are available in claims, in addition to demographics of hundreds of thousands of patients, and decide whether each patient is satisfying all the eligibility criteria or not. For example, in this scenario on the right, you can see a patient with lung cancer that is eligible for a very simple clinical trial looking for patients with any type of cancer. Patient trial matching methodologies can be categorized into rule-based systems and deep embedding-based models. Rule-based systems try to extract name entities and relations from inclusion and exclusion criteria of study protocols and construct rules for identifying patients. These systems mainly rely on huge amount of human annotation or training supervised learning classifiers to extract rules, or a combination of machine learning and rule-based methods. To our knowledge, criteria to query is one of the state-of-the-art end-to-end pipelines which combines machine learning and rule-based methods. These methods generally result in poor recall due to inadequate rule coverage. More recent methodologies use deep embedding-based architectures, which jointly embed patient records and inclusion-exclusion criteria of study protocols in the same latent space and then align them using an attention mechanism. To our knowledge, Compose is one of the state-of-the-art deep embedding-based architectures which utilizes pseudo-Siamese network. On one branch, they have utilized convolutional neural network to learn trial eligibility criteria, and on another branch, they used a taxonomy-guided memory network to learn patient embedding. They also have introduced a composite loss term to maximize similarity between patient records and inclusion criteria while minimizing the similarity between patient records and exclusion criteria. In our work, we are also utilizing a pseudo-Siamese network with highly similar architecture to Compose. In our model, a patient record is a list of claims records which are represented as a sequence of multivariate observations including all their diagnoses, procedures, and products across time dimension. Each patient record is also attached with a set of demographics information such as age and gender. While embedding patient records, we do use multiple levels of descriptions of diagnoses, procedures, and products to account for the medical taxonomy of these healthcare terms. In parallel, a clinical trial record contains of two lists, one of inclusion criteria and the other of exclusion criteria. We are currently ignoring amendments and other modifications to eligibility criteria that are shared as part of a study protocol for simplicity purposes. We then solve for a patient criteria matching problem first, which can be cast as a multi-class classification task, where our goal is to classify matching results between patients and eligibility criteria into match, mismatch, and unknown. And then later we utilize results of patient criteria matching to conclude the patient trial matching decision, our original problem, where a patient is considered a match only if the patient matches all inclusion criteria and mismatches all exclusion criteria. With the model in place, we still need to find ways to address few data and modeling challenges. Some of these challenges are already addressed, and for some of them, we are currently investigating the best methods to address them. First of all, given that the rule-based methods suffer from poor recall, we would like to stay away from generating any and maintaining rules as much as possible. This means we require to have training data for our deep embedding-based architecture. However, historical clinical trial participation is not available to be used as training data for our architecture. And this is due to compliance reasons. As given a clinical trial for a rare disease and few small information about the patient, it is possible to re-identify a patient, which is not aligned with our policies and our goals. Hence, in our work, we use manual data annotation procedures where a team of data experts are helping us to identify anonymized potential patients for upcoming clinical trials. And then we evaluate the correctness of annotated data with clinical trials operations team. Second, even with ignoring amendments and any other modifications that are applied to eligibility criteria, which themselves make things a lot more complex, clinical trial inclusion exclusion criteria can be very complex. First, eligibility criteria often encode more general disease concepts, such as cardiovascular disease, but EHRs represent patients' conditions using more specific medical codes, like PAD, peripheral vascular disease. We are hoping that multiple levels of descriptions that are used per diagnosis or procedures or products can help with this specific complexity. In addition to that, we still need to be aware of criteria that are asking for severity or score calculation at a time of screening, such as the first few lines you see in the math example on the right. Similarly, we can have multi-line criteria where the eligibility decision is applicable when few of the multi-line criteria are satisfied, and there is no need to consider a patient as mismatch if not all multi-line criteria are satisfied. You can see an example of such multi-line criteria at the bottom on right. With such an NLP engine, our vision is to develop a clinical trial decision support system where clinical operation team members can upload their clinical trial study protocols and provide some parameters. They can be related to geographical information, they can be related to their therapeutic area, and then access a list of providers that have potential eligible patients. This vision has been evolving because of our collaboration with clinical trials operation team. With this collaboration, we have multiple goals. Of course, our main goal is to help the business, while we also evaluate the correctness of the input data that we are feeding to our NLP engine. So, following similar manual and rule-based approaches that was discussed earlier, our team is manually identifying providers of potential patients, and then we share that information with the operation team. This way, we empower the targeted outreach program right away. We can also measure the accuracy of our input data and consequently approximate the impact of helping the business with their outreach campaign. Through this collaboration, we are also learning what kind of output really helps the clinical trial team. For example, we can populate a map of number of potential patients of identified providers across the US and also map these numbers for the currently selected sites for a trial and then help identify providers that are within 50 or 150 miles of those sites that are not currently working with Genentech or Roche. And we can also present diversity index data. So the outreach campaigns can be targeted further with respect to some diversity and ethnicity requirements, as you can see in the maps here. In conclusion, we all know that quality healthcare relies on many sources of information, and healthcare professionals are baffled with mountains of healthcare data and expanding medical knowledge that is making it increasingly hard to process and master. We know that clinical operation teams across the US are working tirelessly to advocate for health equity and precision medicine. And we believe AI NLP can help empower inclusive research and facilitate faster research cycles. I hope this talk clarifies the importance of building automated clinical trial matching systems, and I would like to thank you for taking the time to join this talk. Please feel free to reach out if you have any questions, and this is concluding my talk. Thank you.